if we want to improve our quality of life, we need to understand aging at a molecular level. Scientists in the field have worked very hard, and they proposed different drivers or pillars or hallmarks of aging. And the original hallmarks of aging contained 9 and now extended to 12 and even more. But some of the key, including DNA damage, telomere attrition, epigenetic alterations, mitochondrial dysfunction, deregulated nutrient sensing, stem cell exhaustion, cellular senescence, altered cell-to-cell, organ-to-organ communication, loss of proteostasis. So in addition to the original 9, with my colleagues, Mark Madison, David Rubinstein, among the others, we propose that compromised autophagy is an independent hallmark of aging, though there was some overlap between both of them. We also need to keep in mind that all those hallmarks of aging are artificial terms. They are intertwined together. So if in case you are working on DNA damage, it will be also likely to link to many, if not all, the hallmarks we are listed here. When I started my lab in 2017, as you mentioned, we had a small group, so we cannot working on all the hallmarks of aging. So then we wondered whether we can focus on one event that linked to all the hallmarks of aging. And it turns out the small metabolites, Nicotinamide Adenine Dinucleotide, oxidized form, is linked to all the hallmarks of aging, as evidenced by NAD+ reduction can contribute to those hallmarks of aging features, while NAD+ augmentation can alleviate or slow down many of those hallmarks. And with this in mind, there are 3 major topics in my group, including the mechanism of aging, with a focus on the small molecule NAD+. We are interested in why it is reduced during aging and how we can pharmacologically or genetically reinstall cellular tissue NAD+ in a safe way. We are also interested in the aging predisposed dementia with a focus on the dominant one, Alzheimer's disease, with a focusing on garbage recycling or capacity reduction. And we are also interested in collaborating with our colleagues in the using of artificial intelligence as a way to prepare drug development. There are many model systems we can use for aging research. And my group uses C. elegans, which live normally 3 weeks, Drosophila live around 2 months, mice 2 to 3 years, as well as different human samples for our work. So here I want to share with you the project number 1, focusing on defective mitophagy contributed to Alzheimer's disease pathology and is a drug-able target. Excuse me.
I should know her name.
Who is she?
What's her name?
She's looking at you.
I know. I can't remember her name. What's her name?
It's Betty in front of you.
Who are you talking to?
This lady here.
Betty.
I'm Betty.
Well, she's Betty too.
Oh, are you Betty too? Okay. All right. We got that settled. Wait, now I'm gonna get a good night's sleep.
There you go.
Thank you.
The grandma, Betty, she is very sweet and nice, but she is suffering from Alzheimer's disease at the last stage. As you can see, she even cannot recognize herself. And this is a classical feature of Alzheimer's disease. Though there are so many research targeting on this disease, we still do not have a cure. And the population will be increased from around 55 million to over 82 million. So here's the number, those are dementia, with 70% are Alzheimer's disease. We want to give you a little more information on the definition of Alzheimer's disease at a pathological level. The lower part, the right one, is a slide from a cognitive normal person deceased from a disease not related with cognition. And you can see a very like normal anatomic feature. While the left one, a person died of Alzheimer's disease, and you could see shrinkage of the brain structure, especially the cortex, enlargement of the lateral ventricles, and death of the neurons involved in memory and learning, especially in the H region, hippocampus, and the entorhinal cortex. Accumulation of extracellular plaques, especially the A beta plaque, as well as intracellular A beta peptide like 1 to 42, in addition to intracellular tau tangles. Those two are the disease-defining pathological features of Alzheimer's disease. There are many drugs approved by the US FDA as drugs to treat Alzheimer's disease, including the recent approved anti-A beta antibody. But all of them, including the very recent approved one, they can only alleviate the pathology very mildly. So we still do not have a drug to stop the disease. And there's a long way to go. I was trained as a molecular ontologist at NIA. We have witnessed the understanding of the mechanism of aging dramatically over the past decade. There are so many molecular mechanisms like senescence, DNA repair. And we then wondered whether those molecular mechanisms that drive aging can also help us to understand the mechanisms of Alzheimer's disease in view of the statement that aging is the greatest risk factor of Alzheimer's disease. And then we got very precious brain samples from Harvard Brain Bank, and we did a very detailed characterization. The upper one, we finally focus on mitochondria. And the upper one is a patient died of car accident. And you can see that in the memory-related hippocampal region, we see a complete mitochondria with outer membrane crystal inside, although we also see some damaged mitochondria. But compared to Alzheimer's disease patient, the same brain region, we saw accumulation of a dramatic amount of hallowed mitochondria, indicating they are damaged and dysfunctioning. This phenotype was reported for over 20 years, so we are not the first one to show this. And the classical understanding in the field suggesting it could be caused by increased oxidative stress, and which could be also a driver of Alzheimer's disease. But this concept may not be that so prioritized in Alzheimer's disease, as treating the AD patients with antioxidants do not see dramatic benefits. So with this contradictory information in mind, we wondered whether it could be caused by other molecular mechanisms. Before we go to the mechanism, I want to give you more rationale. While when we are working on the brain, we need to focus on mitochondria, because our brain constitutes less than 3% of the body weight, but can consume over 30% of the energy. And the energy correlates like a machine to enable the car, the vehicle to run. And if you look over Betty again, she got married at her 20s. She had a very normal memory at that time. Her husband's birthday, their anniversary, it was when started to reach her 60s, she started to have deterioration of memory. And at her 80s, she was unable to remember the things. And the classical understanding is when a dementia patient was young, they had relatively good neurons, which are able to store the memory. However, when they are getting older, reach to 60s, and even worse, when they are getting older, accumulation of those tau and A beta we consider as garbage could be the driver of the disease. And the concept we proposed is when we are getting older, worn out of the engine can result in reduced garbage recycling capacity. And we aim to repair the engine through a process termed mitochondrial autophagy, mitophagy. There are many approaches can increase our quality of life, potentially reduce the memory loss, such as exercise, fasting, and we are also interested in those processes, but focusing on the contribution of mitophagy in the benefit induced by exercise and fasting. And we're also interested in using mitophagy-specific inducers as an approach to slow down the disease progression, in addition to mental health. So speaking of mitophagy, it is one type of autophagy. While autophagy, I'm talking about the macroautophagy. In addition, we also have microautophagy and chaperone-mediated autophagy. If I know in Boston, it's in the morning or sometime now. If you haven't had your lunch, or if you come here through cycling, rather than taking a bus, you have consumed a lot of energy. So your blood AMP to ADP level is very high, which senses activation of AMPK and shutdown of mTOR. And this initiates the phosphorylation of ULK1 and recruitment of many proteins, followed by the generation of double membrane factor recognize different non-necessary or damaged components, maturation, and finally fuse with acidic lysosome to degrade the components and release them to cells to reuse. There are many proteins involved in this process, and based on the contribution and identification of different autophagy or ATG proteins by the Japanese professor Yoshinori Osumi, who was credited a Nobel Prize in 2016 in medicine or physiology. And here I want to get your attention of only one protein called ULK1, which is important for the initiation of autophagy, as well as participants in the mitophagy pathway. So how to quantify mitophagy? There's a marine protein called KIMA, which is sensitive to pH, while in pH 7 in the normal condition is in green color, while in acidic environment, which is mitophagy autophagy process, then it's in red color. Scientist has expressed the KIMA on the mitochondria. And you can see in the HeLa cell, in physiological condition, we only have less than 1 or 5% of red dots, which is essential, which is enough to maintain mitochondrial homeostasis. However, when the cells are exposed to a mitochondrial toxicant like FCCP, you see dramatic increase of the dots, which are quickly activated to reduce damaged mitochondria, enabling the survival of the cells. And we propose an age-dependent reduction, and especially in Alzheimer's disease, a defective mitophagy could contribute to the disease we are having. Though there were studies showing mitophagy is reduced during aging, but they use primary cells or dead animals. How mitophagy changes in live animal, that was not available to the best of my knowledge. So in collaboration with my colleague Wang Nantan, and we use a two-photon microscopy, and we find that both mitophagy, neuronal mitophagy using the SYN reporter, there was an age-dependent reduction of mitophagy in neurons in the live animals. And mitophagy in astrocytes was also reduced during aging in live animals, with the data for paper in preparation. If we go back to the electron microscopy data, using a two-way blinded manner, we quantified mitophagy-like event, and you could appreciate we noticed 60% reduction of basal level of mitophagy in Alzheimer's disease patients compared to age and sex matched controls. Speaking of molecular mechanisms, we tracked different proteins which were reported for the autophagy mitophagy pathway. And finally, we focused on ULK1, which with a protein level was reduced in AD compared to controls. And in addition, the activity of ULK1 was also dramatically reduced, as evidenced by reduction of serine 555, which is essential for the function of ULK1. And I will focus on this ULK1 in my following project. So then we wondered whether mitophagy is just a bystander if we do not see a driver, but at least a risk factor of Alzheimer's disease. We fed the animals with mitophagy inducer and see whether it was able to slow down the memory loss. And we use different methods for spatial memory evaluation, including the using of Morris Water maze. If any of you haven't used this before, we just put a mouse on this milky water. There was a hidden platform. Here, for the visibility, we put a little bit higher. So the first time it takes 50 seconds or even longer based on how big the trunk is to find the platform. If this mouse is able to memorize, after 5 to 6 times, they can find it in a much short time. And you can see in the left side, which is a demented mouse, even after 6 times training, it was unable to find the target. While the right one, after treated with a mitophagy inducer, it was able to recognize the platform was in this area. And we had around 15 to 16 mice per group covering both genders. And we saw indeed improved spatial memory with our mitophagy inducer treatment. Due to time concern, I want to give you a summary on the project number 1. Using a cross-species approach, we noticed reduced mitophagy in those AD animal and human patients' systems. And this causes accumulation of damaged mitochondria. However, this pathway may not be sufficient to cause Alzheimer's disease. We still need tau and A beta, especially like ApoE4 and TREM2. And they work together to cause neuronal dysfunction and even death, as well as reduced garbage clearance capacity by the supporting cells like microglia and astrocytes, as well as higher inflammation in microglia because of the higher NLRP3 caspase-1 signaling we identified, and finally shrinkage of the brain and loss of memory. So how did we convince ourselves and that reduced mitophagy is at least a risk factor of the disease? Because when we pharmacologically, for example, using the NAD plus precursor Urolithin A as well as genetic approach to reinstall mitophagy, we saw increased mitochondrial homeostasis, increased neuronal survival, increased phagocytotic capacity, and reduced inflammation of microglia, and reduced the tau and A beta, and finally increased memory, as we saw in different A beta and tau mouse models. So I want to share with you a little more on how the Urolithin A is important for mitophagy and NAD for mitophagy. Then next, I want to share with you our understanding on why when we are getting older, mitophagy is dampened. Urolithin A is a small molecule which with its precursor available in fruits like pomegranate. Those polyphenols can be digested in our microbiota and to produce Urolithin A. And our previous results showing UA was able to reduce tau and A beta pathology. And very recently, our following up paper showing Urolithin A was able to regulate the lysosomal homeostasis to increase the degradation of aggregated proteins. And another mitophagy, like another molecule we're interested in is NAD+, which in the Boston region, Professor Nanny Grante, David Sinclair, among the others, are true leaders in the field. So it has been proposed that there's age-dependent reduction of NAD could be caused by increased consumption and or reduced production. If you look over the mitochondrial function, there was also a similar line correlated with aging extension. So I want to give you a little more information on what NAD was discovered and how it is produced in our cells. So it was started by some German professors when they were discussing how fermentation came, and they noticed that a portion now containing NAD+, if without this portion, glucose is unable to be digested to pyruvate. And here, NAD+ to convert it to NADH, I do not use the word degradation. So to convert to reduced form, NADH, and which is used by the last stage, acetaldehyde to alcohol. So NADH to NAD+ again. This explains very well once the fermentation happens in our kitchen, it does not stop. Like you have the pickled cucumber, you should eat this within 10 days, otherwise it will become very sour. And the importance of NAD+, NADH, NADP, and NADPH is coined by credits of 4 Nobel Prizes to 6 professors on their contribution in the understanding of NAD and NADP molecules. It has been proposed that we may die in 20 seconds or within a few minutes if without NAD+ production. I think this statement could come from the importance of NAD in different cellular processes. For example, the glycolysis, glucose to pyruvate, which needs NAD+. Pyruvate enters into the matrix of mitochondria for TCA cycle, a process which also needs NAD+. When we eat fat, which degrades to acetyl-CoA, which also uses NAD+ through the beta oxidation process. And NAD+ in those through process to NADH, which is used by the mitochondrial complex, especially complex 1, to generate 30 copies of ATP for us to use. And to understand why when we are getting older, NAD is reduced, we should understand how NAD is produced in mammals. At least there are 3 to 4 molecular mechanisms on NAD+ production, including the de novo pathway. De novo pathway cells use tryptophan to generate NAD+, which is a very complicated, time-consuming, and less efficient approach. But many of the intermediates inside are very important if you are working on neuroscience or in the neurology department. Those intermediates are with the blue asterisk, like hyaluronic acid, picolinic acid. Those are neuronal protective, while 3-HA, 3-HAA, and culoninic acid are neuronal toxicants. The second pathway, cells use nicotinic acid to generate NAD+, which is a far more convenient and efficient way. However, many of the patients suffer from this itching and flushing side effects. And the third pathway is NAD to be degraded to nicotinamide, and which finally converts to NAD+. Charles Brunner in 2004 or 6 showed that nicotinamide riboside NR is another precursor. So all those pathways together we consider as salvage and recycling. The current understanding of the benefit of NAD in longevity could contribute by its role in DNA repair, to be used by the pups to repair damaged DNA, to be used by the NAD-dependent sirtuins, which can deacylate proteins like foxols, GC1 alpha, to increase a transcriptional overexpression of genes involved in metabolism, neuronal plasticity, healthspan. Also, the role of sirtuins in longevity with some controversial, but its benefits in healthspan is very solid. There are other molecules involved in. So if you are in the NAD field, there are different precursors are used, including nicotinamide riboside, as well as nicotinamide mononucleotide. And my group use all of them, and majorly NR and NMN. I just want to share with you that when we giving NR or NMN or ND plus to humans, the majority is digested via microbiota with a small portion of NR and majorly NAM and nicotinic acid to be consumed and absorbed in our body. With our colleagues, we show that carnitinic acid, which in the de novo pathway, if this molecule is higher in CSF, it can slow down the deterioration of memory in a longitudinal cohort we worked in Norway. And if any of you know the disease delirium, or in Chinese, it is Zhangwan. So this happens very often when you get hip fracture or COVID-19 patient in ICU. And we showed that if a patient has high levels of quinolinic acid, which is intermediate in the de novo pathway, then this individual has a high chance of death compared to others. And how NAD induces mitophagy? The pathway we contributed was that when we increase NAD+, it increases the deacylation activity 31 to increase the deacylation of FOXO3, which can transcriptionally upregulate genes involved in autophagy and mitophagy, such as BNIP3 and LC3. And there are many other molecular mechanisms. I won't go to details. And in Norway, we are very strong in outdoor activities, especially skiing. And when we started the work, and my students were asking, like, in addition to fed the mice with NAD+, if we also force them like to run like through the treadmill, and we indeed saw synergistic benefits to different parameters we have been working on with the paper in preparation. And here I want to go to the next one to explain why when we're getting older, more or less, we have impaired memory, even we cannot reach to Alzheimer's disease. So as I showed you, the autophagic process with ULK1 involved in the initiation of the autophagy pathway, which is also important for the mitophagy pathway, including the pink-1 parking dependent as a downstream, as well as important for the pink-1 parking independent pathway, like FU and DC1 pathway. So we wondered whether there's any linkage between Alzheimer's disease and mitophagy in collaboration with Fei-Sheng Chen from Cleveland Clinic in the USA. And we found that ULK1 protein is indeed clustered between the two features. And then working with our colleagues in University of Oxford, we worked on different autophagy, mitophagy, mitochondrial pathway-related proteins. And we found that compared to control, indeed, those proteins are either increased or decreased in Alzheimer's disease. ULK1 is not in this list because it was not in the soma scan antibody-based screening method they used. It does not mean it was no change or undetectable. We showed before that ULK1 was reduced in protein level as well as in activity. So we wondered whether this is a coincidence or it is a case. And by using a different longitudinal cohort in Norway, including a healthy population, older population, after 4-year follow-up, as well as in cognitive normal compared to Alzheimer's disease, including mild cognitive impairment and Alzheimer's disease, as well to use Alzheimer's disease brain samples. 5 to 6 are severe stage, 3 to 4 are early stage of Alzheimer's disease. Those are control. And by using all those systems, we show an Alzheimer's disease and aging-dependent reduction of ULK1 in both serum and CSF. And then we wondered whether ULK1 reduction could contribute to memory loss. And what we did was we overexpressed ULK1 in both A beta and tau mouse models. And we indeed show when we overexpress of ULK1, either ubiquitous or neuronal specific, we saw increased mitophagy, increased autophagy, reduced damaged mitochondria, and improved cellular function, survival, and increased memory. With this paper also in revision. Because we had this ULK1 model, so when our students was maintaining the colony, they noticed that ULK1 overexpression compared with wild type may live longer. So then we had 2 sets of serious experiments checking the lifespan. And you can see that indeed, male or female, I think on average, ULK1 overexpression reached 25% extension of lifespan, especially in females compared to male. And we had 2 biological repeats. It seems ULK1 overexpression not only makes the animal live longer, but also healthier. By using this frailty score, which with like gray hair and behavior, total 31 parameters, the higher score indicates the higher frailty. If you look over the pink color, there was an age-dependent increase of the parameter, while ULK1 overexpression continuously and consistently reduces those parameters, indicating improved health span, in addition to other parameters I didn't show you here. So we also have unpublished data showing ULK1 overexpression can reduce depression in mice. Here I want to go quickly because I do not have much time on the AI-based project, just to give you a summary. I want to use the time to explain on how important AI in our daily life, in addition to dramatically facilitates medical care and disease identification, and even for clinical trial. But I want to give you one slide to summarize a paper we published a few years ago. So back to 2019, we're interested in identification of physiological on those safe mitochondrial inducers as potential drug candidates. And during that time, there was only 19 mitochondrial inducers which can induce mitophagy through molecular mechanisms not to damage mitochondria. Like we excluded FCCP rotulon inside. So we had a total of 19 positive components, and we collaborated with our colleagues in Imperial College London in the UK, and they built an algorithm for us through these 3 methods after a training of an online big database. Then we went access to a small natural compound library in the University of McCall, which contains over 3,000 compounds identified from traditional Chinese medicine with a history in treating memory loss, and it showed positive results before. So we wondered which molecules can induce mitophagy and can also potentially reduce the memory loss. First, we tested in cell culture like HeLa, this video I showed you model system, with a 10 micromolar as a cutoff dose. 8 of the molecules passed this stage of the criterion. Then we wondered in the C. elegans model of Alzheimer's disease as well as mitochondrial inducer. We wondered whether those 8 molecules was able to increase neuronal mitophagy and reduce memory-like deterioration. And we saw 2 passed these 2 criteria. After that, the 2 compounds were subjected to 3XAD mouse model, which harboring both tau and A beta pathology. And very luckily, those 2 molecules were able to increase mitophagy in the brain and reduce the memory loss. And those are our lead compounds. And now we are doing further structure modification to increase their specificity in inducing mitophagy. And here I want to show with my product number 4, that is identification of a new mitophagy induced in passion fruit. And my student Shu Qingcao, when she started her PhD in Thailand, a country they eat a lot of colorful fruits. And there are many epidemiological studies showing eating more colorful fruits correlates with low chance of dementia. So she wanted this was a coincidence because those people have better salary and better access to healthcare, or there are molecules inside can reduce the memory loss. And Shu has identified more than 100 metabolites, small molecules. And finally, we focus on the EFFAA, which seems to be one of the contributors. So first, we showed that I want to go to details, but to show when we we inject the molecule into the mice, there was a dose-dependent induction of mitophagy using a MitoQC, which is also a mitophagy reporter. And it seems this pathway could be at least contributed by the Pink1-Parkin-based mitophagy by using this phosphorylated ubiquitin and serum 65 as a marker. And we also show this EFA molecule was able to improve the memory, reduce the tau pathology, especially p-tau217, which is the recently identified p-tau, which can be used for early detection of memory loss and a specific marker for Alzheimer disease. And this molecule is also able to reduce damaged mitochondria and increase mitophagy. In addition to the studies in mice and C. elegans, we wondered whether our molecule is able to improve function in the organoid model by using iPSC to differentiate to neurons, astrocytes, and then added microglia in red color at the end, in collaboration with our colleague Hansen Cho, who also got his postdoc training in Harvard Medical School. And you can see that using this AD model, it recapitulates the tau pathology, while the wild type had very low p-tau217 and AD had a very high level. And with our molecular treatment, it can dramatically reduce the level. And the migration of microglia in red color from the rim to the center indicating inflammation. And our molecule can dramatically reduce inflammation. And further, we wanted to check whether this molecule what are the pharmacokinetics. And our molecule, the half-life in the blood of mice is around 10.1 hours, so which is a favorite time if you want to develop a drug. And this molecule is also able to pass the blood-brain barrier with experiment done by a commercial company in the Netherlands. And we had the crude extract of passion fruit, and which shows improved memory in C. elegans. And we got some news from the national media of Norway. And I remembered this news was published on Friday. And on Saturday, I went to the market to buy a passion fruit, which is my routine. And you can see what happened. The passion fruit was gone. And my colleague told me all the Norwegians follow the signs from the national TV. So it also makes us to be more responsible in our data interpretation. And I want to share with you that in Norway, we have so many high quality resources like the sea, fish and shrimps there, including this krill, which is only available in the South Pole. And Norway has a region there to be qualified them to hunt. And a few years ago, with my head of department, Hilda Nielsen, we showed krill oil was able to increase resilience of Parkinson's disease harbored neurons and reduce the biological age and extends the lifespan and health span in C. elegans animals. Even we do not know the results in humans, but the company, many companies did studies in humans showing positive benefits. So this is very exciting. And with all those in mind, in collaboration with my co-former mentor, Mark Madison, and my long-term collaborator, Professor David Sinclair in Harvard. We wanted to ask why, when we're getting older, like 70 to 80 years old, we more or less have memory loss, but only 10% or less than those percentage can get Alzheimer's disease. So how to make this thing happen in a molecular mechanism-based way? We propose there's an age-dependent change on NAD mitophagy pathway. When we are young, this pathway is good. We have a high quality of mitochondria, high capacity to reduce garbage. However, when we're getting older, NAD is low, mitochondria is dampened, and the recycling is done. However, this may not sufficient to make somebody get Alzheimer's disease when we're 18 years old, 80 years old. We still need ApoE4 and TREM2, similar for Parkinson's disease and Huntington and ALS. In view of the importance of autophagy in safeguarding quality of life, with Guido Kromer, Linda Patrick, David Rubinstein, among the others, we propose compromised autophagy is an independent hallmark of aging, which was also applied to their 2023 updated hallmarks of aging in Cell. We are not only just publish a paper, we really want to see whether our work can be applied to the clinical setting. In collaboration with our colleagues in Charles University in Prague, we asked whether any of those mitophagy autophagy proteins in the blood can be used as a biomarker to track the progression of the disease. We collected 46 control, 100 mild cognitive impairment, and 100 AD, and we found the pink one was increased when AD happens. NIX was also high, while TFAB was low. We don't know why. Some are high, some are low, but those are the results we got. And in Norway, I'm very fortunate to be involved in many clinical trials, such as for using NAD to treat ALS, using NAD to see whether it can change, can alleviate side effects for the long COVID patients. If any of you are interested in AD, there's also a big discussion on NAD and cancer. So while whether NAD can increase or reduce cancer, both data are there. But before we have conclusion, we should be cautious if any of us have a cancer. It's not suggested to have NAD until further evidence. I'm very pleased to build this Norwegian National Anti-Alzheimer's Disease Network with many colleagues on board, including our Harvard colleague Brad Hyman, MIT Professor Li Huicai as our board members. We also built this Norwegian Center on Healthy Aging network with many of the colleagues in the field trying to working together to fight against aging. And finally, I want to share with you if any of our family members or somebody got Alzheimer's disease, we should encourage them to go outside rather than to stay at home. A recent TV documentary by our national channel in Norway showing that if we let the patient to go seeing once per week, they can have dramatic benefits to the life and reduce the memory loss. And this documentary has got many awards, including the Human Rights Awards by the University of Oslo. And very recently, we got a big funding to investigate whether and how delirium as a contributor or risk factor of Alzheimer's disease. And I'm very pleased that even we have this lab for less than 7 years, but our work has been recognized by the leaders in the field. So we really thank the students in our group. And finally, I want to give you a summary of today's work. I didn't show you some of the results, but in a group of premature aging disorders, DNA repair deficiency can cause NAD reduction as a way to exacerbate the aging process. The NAD mitophagy process is essential for healthy longevity, while it is dampened in aging and extremely degraded in Alzheimer's disease. Defective mitophagy is a risk factor of AD, while mitophagy is a drug-able target. ULK1 is reduced in Alzheimer's disease, while its augmentation forestals AD pathology and retains memory. AI could be used to screen drug candidates for Alzheimer's disease, and we have identified a novel mitophagy inducer EFA passion fruit with anti-AD potential. And many of the autophagy mitophagy proteins are detectable in blood samples, and there's a correlation with the Alzheimer's disease conundrum. So how we can improve our quality of life? I think many of us are molecular gerontologists working at a molecular level. So while we are targeting different hallmarks of aging, we also which contribute to different major aging-related disorders like cardiovascular disease, osteoporosis, among the others. We also need to consider how to improve the aging by intervention at individual level. With many of the approaches we have, like solutions we propose as right side. At the societal level, we need to talk into the government, the policy makers, how to build older people friendly environment with our suggestions here. So recently we have made this strategy to launch healthy longevity.